Welcome back to the Deep Dive, where we cut through the noise of complex research to bring you the core insights. Today, we are going deep into a field that sounds like science fiction, but is very much a reality: tissue engineering. And we're going to focus on really the absolute foundation of it all, the materials, the scaffolds that are used to literally regrow human tissue. Our sources for this are a number of intensive academic analyses looking at the biomedical engineering that makes these devices possible. And to guide us through this incredible intersection of biology and material science, we are so honored to have technologist Dr. Rama Yusvana with us. He's a senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, or UTHM.
It's a real pleasure to be here. This is, such an exciting field, and you're right, it all comes down to the materials. Get that wrong, and nothing else works.
So let's start there with the problem that even created this field. For centuries, really, doctors have had a plan A for damaged tissue. What were those conventional treatments, and I guess why aren't they enough anymore?
Well, those conventional treatments have been and still are life-saving. We're talking about two main types, autografts and allografts.
Okay, so what's the difference?
An autograft is where you use the patient's own tissue. Think of a skin graft from one part of the body to another. An allograft, or a transplant, is when you use tissue from a donor.
Right. And they've been revolutionary, but they have limits.
They do. Significant limits. And that's really what pushed science towards regenerative medicine.
Let's take the autograft first. Using your own tissue? It seems perfect. Your body isn't going to reject it. So what's the catch?
The catch is twofold. First, you're limited by supply. You only have so much spare skin or bone to harvest.
So of course.
But more importantly, you create something called donor site morbidity. To fix the problem you have here, you have to create a new wound over there.
You're creating a new problem to solve an old one.
Precisely. That harvesting process, it's painful, it's expensive, and the new wound site is now at risk for things like infection or, a hematoma. It's a serious trade-off.
Okay, so if autografts are limited, that naturally leads to allografts using donor tissue. But that opens up a whole different can of worms, doesn't it?
A huge one. Logistically, there's just a massive scarcity problem. Not enough donor organs or tissues to meet the global demand.
Right.
And then biologically, you have the big one, immune system rejection. The body sees that donor tissue as foreign and attacks it.
Which means a lifetime of immunosuppressant drugs.
Yes, which have their own health risks, not to mention the possibility of transmitting diseases from the donor to the recipient.
So that sets the stage perfectly for tissue engineering. The idea is to just bypass all of that.
Exactly. The goal isn't replacement anymore, it's regeneration. You're creating the conditions for the body to heal and rebuild itself.
And that takes a whole team, right? This isn't just one discipline.
Oh, absolutely. It's a convergence of clinical medicine, material science, genetics, all working together.
So let's talk about the core mechanism. How does it actually work? What are the key ingredients?
It all comes down to what we call the tissue engineering triad.
The triad. Okay.
You need three things: cells, often from the patient, a scaffold, which is a biomaterial, and then signals.
Let's break that down. The scaffold, what is that exactly? Is it like the scaffolding on a building?
That's a perfect analogy. It's a temporary, highly porous structure, a biomaterial, either synthetic or biological, that we implant. It provides that initial structural support, and it acts as a template, guiding the new tissue into the right shape.
So it tells the cells where to go.
And it helps transport nutrients to them while they grow. It's their temporary home.
And the third part of the triad, the signals, what are they telling the cells to do?
The signals are the instructions. They can be biochemical, like growth factors that we embed in the scaffold, or they can be biophysical stimuli, maybe from a bioreactor in a lab.
A bioreactor.
Yes, a device that can simulate the body's environment, maybe applying pressure or flow to encourage the cells to organize correctly. These signals tell the cells to grow, to differentiate into bone or cartilage, and to start building their own matrix.
So does this all happen inside the body, or do you build the tissue in a lab first?
It can be either. We can seed the cells onto the scaffold and culture it in vitro in the lab to grow a piece of tissue, and then implant it.
Okay.
Or we can implant the scaffold directly in vivo and rely on the body's own cells and signals to populate it and do the work. Either way, the engineering of that scaffold has to be incredibly precise.
I can imagine. It has to be strong, but also temporary, non-toxic.
Yeah.
It's a huge list of requirements. Our sources mention 6 key pillars for designing one.
That's right. For any tissue, you have to get these 6 things right: biocompatibility, biodegradability, mechanical properties, the scaffold architecture, the manufacturing technology, and finally, the choice of biomaterials.
That is a lot to balance.
It is. And the fabrication method you choose, whether it's something like, 3D printing or electrospinning, often depends on which of those 6 pillars is the most important for that specific application.
Let's dig into the bioproperties first.
Definitely.
Biocompatibility. That sounds like it just means not toxic, but it must be more complicated than that.
It is much more. Not toxic is the bare minimum. True biocompatibility means the material has to be a perfect host for the cells.
A perfect host.
Yes, the cells have to want to attach to it. They need to be able to migrate through it, to multiply, and then most importantly, to start producing their own tissue matrix to replace the scaffold.
And what about that big enemy from transplants, the immune response? How does a synthetic material avoid triggering rejection?
The goal is what we call a negligible immune reaction. You can't have a major inflammatory response. If the body starts trying to attack or wall off the scaffold, the whole healing process fails.
And that inflammation could cause other problems, right?
Absolutely. Our sources point out that chronic inflammation is linked to all sorts of serious conditions, like atherogenesis, the plaque buildup in arteries. So a poorly designed scaffold could cause systemic problems far from the implant site.
Wow, okay, so it has to be biocompatible. Then there's biodegradability. Why does it have to disappear? Why not just leave it in there?
Because it's only a template. It's not a permanent implant. The whole point is for the body's own tissue to take over. If the scaffold stays there, it gets in the way. It prevents the formation of a fully functional natural tissue.
So the timing has to be perfect.
Perfect. It's maybe the biggest challenge. The scaffold has to degrade at the exact same rate that the new tissue is being formed.
What happens if that timing is off?
If it degrades too quickly, the structure collapses before the new tissue is strong enough to support itself. Degrade too slowly, and you're left with foreign material blocking the final stages of regeneration.
And what about the stuff it breaks down into?
Also critical, the byproducts of that degradation must be completely non-toxic and easily cleared by the body, without putting any strain on the kidneys or liver. It requires a huge amount of chemical and immunological study.
Okay, that covers the biological side. Let's move to the physical, the mechanical properties. This must be where one size definitely does not fit all.
Not at all. The scaffold has to match the mechanical needs of wherever you're putting it. A scaffold for bone needs to handle compression. One for skin needs to be flexible and elastic.
And it has to survive surgery in the first place.
That's the first test. The surgeon has to be able to handle it, suture it, and place it without it just falling apart.
Where are the biggest mechanical challenges? Which tissues are the hardest to replicate?
Definitely the load-bearing tissues. So orthopedics, bone, and cartilage, and cardiovascular applications are incredibly difficult.
Because they need to be really strong.
Exactly. They need to withstand the body's forces from day one. But here's the paradox. To be strong, you usually want a dense material. But for tissue to grow, you need it to be highly porous.
So cells can get in.
Right. For cells to infiltrate and for blood vessels to form what we call vascularization. Strength and porosity are almost always in opposition. Finding that perfect balance is, well, it's the holy grail of scaffold engineering.
That brings us perfectly to the next pillar, the scaffold architecture. So it's not just about having holes, it's about the right kind of holes.
Precisely. It's about having a highly porous, interconnected structure. That's the non-negotiable part.
Interconnected, why is that so crucial?
Because it creates channels. It governs the whole exchange of life. It ensures nutrients, like oxygen, can get deep into the structure, and it ensures waste products can get out. If you don't have that flow, you get a big problem, especially in thicker scaffolds, called core degradation.
Meaning the middle dies off.
The cells in the very center essentially suffocate and starve because there's no way to get supplies in or waste out. The vascular system can't penetrate a dense core.
So the architecture is essentially the plumbing system for the new tissue.
A perfect way to put it. And the surface chemistry of that plumbing matters too. Cells interact through specific chemical groups on the surface called ligands.
Okay, and do all materials have these ligands?
Not equally. Natural materials, like collagen, have them built in. Cells are naturally attracted to them. For many synthetic materials, we actually have to engineer the surface to add these ligands on so the cells will recognize it and attach.
That creates another balancing act, doesn't it? The pore size.
A very delicate one.
Oh.
The pores have to be big enough for cells to physically move through them.
Right.
But if they're too big, your total surface area inside the scaffold drops. You need a high specific surface area to have enough of those ligand binding sites for a critical number of cells to attach. too big, and they can't find a foothold.
So there's a sweet spot.
There is, and it's different for every cell type. Bone cells, osteoblasts, have different needs than skin cells. It's an incredibly complex optimization.
This all sounds amazing in the lab, but how do you translate this to a real product a doctor can use? That brings us to manufacturing technology.
And the keywords here are scale and standardization. What you can make once in a lab is very different from what you can produce reliably for thousands of patients.
It has to be cost-effective.
And it has to meet very strict good manufacturing practice, or GMP, standards. The process has to be robust and reproducible every single time.
And what about for the clinicians themselves? What do they want?
They want off-the-shelf availability. Ideally, they want to be able to just take a scaffold out of a package and implant it. They don't want to have to wait weeks for cells to be cultured in a lab. That's not always possible, but it's the goal.
So even with the right design and manufacturing, it all still comes down to that initial choice of biomaterials. Let's use an example. What about ceramics for bone? They seem like an obvious choice.
They do on the surface. Materials like hydroxyapatite, or HA, chemically look a lot like the mineral in our own bones. They're biocompatible, they encourage bone cells to grow, and they're very stiff.
But there's always a but.
The but is their physical properties. They're very brittle, they have almost no elasticity, so they can fracture easily, and they're hard to shape into complex forms.
And does the new bone that forms in them work properly?
That's the biggest issue. It often doesn't. The new bone tissue that forms inside a ceramic network often can't handle the mechanical loads needed for the body's natural remodeling process.
Yeah.
So you get bone, but it isn't fully functional bone.
So if single materials always have these trade-offs, what was the next step?
The next step was composite scaffolds.
The same material.
Exactly. You take the best properties of each. You might take the stiffness of a ceramic and combine it with the elasticity and, the biological friendliness of a polymer.
Can you give us a concrete example of that synergy?
A great one is collagen and hydroxyapatite.
Yeah.
A pure collagen scaffold has amazing porosity. It's great for cells getting in, but it's mechanically very weak, like wet paper.
Okay.
A pure hydroxyapatite scaffold is strong, but cells struggle to get inside it. But if you make a composite, collagen HA scaffold,
You get the best of both worlds.
you get the best of both. You get the high porosity and the bio-friendly nature of the collagen, but now you have these HA particles distributed throughout, providing the mechanical strength it was missing.
But you still have to watch out for issues with biocompatibility.
Always. Every time you introduce a new material into the mix, you have to reevaluate all 6 of those pillars again. It's always a balancing act.
This has been just an incredible look into, well, the blueprints for life. We started with the limitations of just moving tissue around, the pain, the rejection, and we've ended up with this idea of an exquisitely engineered structure, one that has to be strong enough for a surgeon to handle, porous enough to breathe, and chemically attractive to cells. And on top of all that, it has to be designed to fail perfectly, to dissolve away at the exact moment its job is done, allowing new, living tissue to take its place. The sheer complexity of engineering a material that has to succeed by perfectly timing its own destruction, all inside the human body. It's just a profound thought. And for that, we have to express our utmost gratitude for guiding us through this. Thank you to our guest, technologist Dr. Rama Yusvana, a senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, UTHM, for his enlightenment and his invaluable insights.
Thank you. It was a fascinating discussion.
And thank you, our listener, for joining us on this deep dive. We'll see you next time.